Good afternoon, Amrita.
Good afternoon, sir.
Let's wait for others to join.
Yes, sir.
Okay. Why are some people leaving? And Adish left, Shivani left. Okay. Okay. So, we covered, how to make a library, gene library, right? It could be a cDNA library or a genomic library. And typically, when it comes to prokaryotes, we would make a genomic library by virtue of the fact that there are no introns, and a genomic library pretty much represents the entire genome, right? And we also had some calculation. We made some, how many clones to be screened if you are looking at a genomic library, right? cDNA libraries are typically used for eukaryotes where there are lots of introns, right? We typically took an example of human genomic DNA and this thing, human cDNA libraries, right? You would use the human genomic DNA library when you want to know, for example, how is this particular gene being controlled? You want to understand the promoter structure. You want to understand, what are the protein factors or transcription factors that are binding upstream of that gene promoter and how is that being regulated. You would probably be looking at genomic library. Otherwise, if you are looking for a homolog, then cDNA library is the best way to go. We also looked at how plasmid vectors can be used, but it is so tedious because you'll have to screen literally millions of clones. Instead, you can use phages as vectors because lambda phage you can remove at least up to 20 kilobase pairs. The region that is responsible for lysogeny can be safely removed, and you can put your gene of interest, and it can tolerate at least up to 40 kilobase pairs. Anywhere between 20 to 40 kilobase pair gets accepted by lambda. And then you have bacterial artificial chromosomes, yeast artificial chromosomes, and several other artificial chromosomes. Okay, all said and done, beautiful. You have made a library and you want to screen now, right? Remember the MIT lecture where they talked about functional complementation. You basically would take a mutant, say, let's say an E. coli mutant, which cannot utilize, let's say, histidine, and then you transform that with the library, genomic library. All you need to do is to plate them on minimal media without histidine, and if any gene fragment that can restore this function of utilizing histidine, those colonies will grow, and you can go back and find out what is that piece of gene, what is that piece of DNA. By sequencing you will know. Of course, as I said, some of these are historical techniques, right? Abhi, the technology is sufficiently advanced that nowadays people first sequence, right, put it in the database, and then they slowly start unraveling these things. Okay. Now, assuming that there is a gene that you would like to look for in your new library, people used to use, various hybridization techniques, right? People used what is called as a southern hybridization. People used northern hybridization. People used also western hybridization. There is a beautiful reason why it was called as northern and western, simply because when there was a scientist by name Ed Southern. Okay, he was the one who first you know, I should say, invented this technique of, seeing. Suppose, you have made a library of a novel organism, right? And you are looking for, let us say, any gene, particular gene. Say, let's say you are looking for a ligase gene or a polymerase gene, or it could be a gene that's responsible for, let's say, lactose utilization. Okay, what you have in your hand is, say, a lag gene, or the any other gene, polymerase gene, or some ligase gene from some other organism, related organism, but not the same this thing. Okay, so all you need to do is to use these different enzymes that we talked about in the enzymes in molecular biology class, and generate radioactive probes, which we will talk in tomorrow's class. I think either tomorrow's class after the day after one. Okay, so you generate radioactive probes. Okay, and then you go ahead and do what is called as hybridization. What is the principle of hybridization is simple. Two strands, each of the two strands of DNA are complementary to each other, right? Suppose in a big pool of library, okay, you are looking for, let us say, a ligase gene, okay, and somebody has donated to you a part of the clone. Say ligase gene, let us say, is about 1 kilobase pair, and they've given you about 750 nucleotides. That is what they had. So they gave you that piece of DNA. Okay. All you need to do is to take this DNA, which somebody has donated to you, make radioactive probes, and use these probes to hybridize. Okay, all these sequences will go and hybridize very specifically only to ligase gene, because the ligase gene sequence is unlike any other gene sequence. The ligase gene sequence does not resemble polymerase gene sequence, does not resemble the LAGZ gene sequence, does not resemble any other gene sequence, right? So this is going to have a very specific hybridization. Okay, and if you take closely related species, it's highly likely, right? For example, if you take E. coli DNA polymerase and you are trying to make a probe out of that, and you are trying to hybridize it to a say another bacterial genome, there is a high likelihood that you will have at least anywhere between 70 to 90% homology, at least 70 to 90% similarity, right? If you take E. coli DNA polymerase and human DNA polymerase, probably about 20 to 30% similarity or homology, right? And if you take E. coli DNA polymerase and yeast DNA polymerase, for example, you may get up to about 50-60% homology, right? But closely related species, the closely related species will have very high homology, and the more distant they are going to have less homology, right? So that is the principle by which hybridization techniques work. Today we will look at three techniques. We will have an overview on southern blot. We will have an overview on northern blot. We will have an overview on western blot. Right? You may have had these lectures already, or maybe you're getting exposed to this for the first time. I just think that pay close attention. You will know that there is not a whole lot of difference between northern and southern blot. Northern blot is not, southern blot is nothing but DNA hybridization. Okay, and everything else remains the same for northern blot. Instead, the probe is used. The probe that is used for northern blot is derived from RNA. Okay, so essentially you are doing a RNA DNA hybridization. RNA DNA hybridization is called as northern blot. Okay, DNA hybridization is called as southern blot. Southern blot was the one that was first invented. Okay, and because the scientist's name, as I said, was Ed Southern, he called it a southern blot. And because he called it a southern blot, somebody said, I will call this as a northern blot. Okay, it just out of this thing. So they called it as northern blot. So northern blot is nothing but RNA DNA hybridization. The moment you had southern and northern blot, somebody did what is called as Western blot. Western blot is nothing but antigen antibody hybridization. It has nothing to do with nucleic acid. It has nothing to do with nucleic acid. So when it comes to nucleic acid hybridization, it is DNA or DNA RNA. These are called as southern and northern blots respectively. Western blot is nothing but hybridization or the recognition of an antigen and to antibody. Okay, that is what is called as a Western blot. Okay, so let's quickly, have an overview of all these three techniques, and then we will probably, look into the. Let's have the principles of this, and then we look at each of those things in a little more detail. Okay. So let me share my screen. All right. I think the screen is visible now. Let's. So as I said, Western blotting typically involves proteins and antibodies, right? Antibodies and antigens. Southern blotting involves DNA and DNA. Northern blotting involves DNA and RNA. Okay. So let me put this on the full screen mode.
To speak about southern blotting and northern blotting. Why? Because simply the principle of these two is very, very similar. So basically they are the same technique, but with like some differences, because this one is used for DNA and this one is used for RNA. So let's start first speaking about southern blotting technique. As I told you, southern blotting technique is used to detect specific sequence of DNA. So saying that I have a cell, eukaryotic or even prokaryotic cell, and then I extract the genome from this cell. So
it's very important to understand whether it is southern blotting or northern blotting or western blotting doesn't distinguish between eukaryotes, prokaryotes, and all that. Okay, so southern blotting is used equally for prokaryotes and eukaryotes. Similarly, northern blots can be used equally for prokaryotes and eukaryotes, right? And western blot similarly is useful both in prokaryotes and eukaryotes. For example, let's say you have a piece of DNA, somebody has donated you, as I said, ligase gene, and you want to know whether human has human genome as ligase gene or not. Human genome has tons of ligase genes. I think at least there are 8 different ligase genes. Okay, so all you need to do is do a southern blot, and you will know whether the human genome has ligase gene or not. Okay. Similarly, suppose you know all of you are now very well aware of the corona pandemic and the COVID surface antigen, right? So if you have raised antibody to that corona spike protein, and suppose you want to know, whether the spike protein is present in any other organism or similar to spike protein if there is anything present in other organism, all you need to do is take this antibody against the spike protein and take the test organism. It could be a bacteria, it could be a virus, it could be any other test system. Run the protein gel and then use this antibody to go and find out if they have a corresponding spike protein or not. Because of the fact that these antibodies are highly specific, right? They're highly specific to spike protein, right? If there is a spike protein like this thing in your test organism, it will go and bind. If it doesn't have, it will not bind. Simple, all these hybridizations are very, very specific, okay? But at the same time, you can also tailor conditions so that you can have low stringency. You can have a highly stringent hybridization. That means only when there is a perfect this thing, interaction, you will see the signal. If not, you may not see the signal. Right? At the same time, you can have a low stringency. Okay, that means your hybridization conditions are not very strict. Okay, in that case, you will see certain, loosely hybridized this thing. You can. That is also a very useful technique. Okay.
So I have a genome sample. The thing is that I am interested in searching about a specific sequence in this genome, maybe a specific gene or a specific sequence I want to search about in this genome, sorry. What I should do first is that I should perform agarose gel electrophoresis. So step 1 in Southern blotting is agarose gel electrophoresis. I also have there is a previous video in my channel talking about agarose gel electrophoresis. So if you didn't watch it, I also recommend you to go watch the agarose gel electrophoresis video. Now, first of all, the genome or the DNA sequences should be separated using agarose gel electrophoresis. We use a couple of enzymes called restriction nuclease enzyme. The restriction in the nuclease enzyme is going to cut the DNA in different places and is going to produce several sequences of the DNA of the genome. The thing is that to decide which endonuclease enzyme to use, I should know the sequence I have. Why? Because as I told you in the agarose gel electrophoresis video, the restriction endonuclease enzymes are many. So there are a lot of restriction endonuclease enzymes, and each one of them detect the specific sequence on the genome and cut the DNA downstream this sequence. So in order to choose the restriction enzyme I want to use, I should know the sequence of the gene of my interest. After digesting the DNA or the genome, I apply this sample on the gel, on the agarose gel electrophoresis, using agarose gel and an electrical current. Then the DNAs will be separated. Of course, I might have several samples, and then the DNA sequences will be separated according to their length. The thing is that in order to search about a specific sequence, I should perform Southern blotting. Why? Because if I visualize the gel like this, I will see the gel in this way. I told you about visualization in the previous video about agarose gel electrophoresis using ethidium bromide, cybergreen, or other molecules. I will see the gel like this. So the thing is that I cannot differentiate the sample of my interest from other samples because I'm interested in only one sequence. So I cannot differentiate the sequence of my interest or the gene of my interest from other genes. And to do so, I should perform Southern blotting. Southern blotting, let's say I have this gene, I'm interested in visualizing this gene. So first of all, I should know the sequence of this gene. Let's say this is the gene of my interest. I should know the sequence of this gene, and then I should design a complementary sequence of this gene. So like this, for example, let's say AAGGTCTT, then I should design a complementary sequence. Because it's complementary to it. But how can I perform this? In order to perform blotting, what I should do is that I should transfer the sequences in the gel or the samples of DNA which are in the gel to a membrane, a nitrocellulose membrane or another type of nylon filter membrane. Why should I do so? Because when I transfer the DNA samples on the membrane, the DNA samples will bind on the surface on the membrane and then the probe can detect the sequence and bind to it, which cannot be done in the gel. Now, how can I perform the transfer step? In order to transform the transfer step, first I should use a chamber like this, filled with the buffer. The buffer is an alkaline buffer. Then I use a sponge. So this is a sponge. Then I put my gel. This is the gel which contains the DNA samples inside. Then I put the membrane on a direct contact with the gel. Then I use paper towels, and then I use a weight, a kind of weight, in order to compress all the stuff together. So what I have here is that I have the alkaline buffer, I have the sponge, the gel, the membrane, DNA binding membrane, the paper towels, and then a weight. What's going to happen then in this orientation is that I will have the bottom, which is very rich in water or very rich in the buffer, and then it will in this part there will be high water pressure, and in this part, which is totally dry, there will be low water pressure. Because of this, there will be a kind of capillary forces which is going to drive the water or drive the buffer from the area of high water pressure to the area of low water pressure. So it's going to move the water or the buffer against the gravity from the bottom toward the top. Now what's going to happen is that when the buffer is passing through the gel, it's going to take the DNA samples with. But then when the water is going to pass through the membrane, the DNA samples will stick on the membrane. Why? Because of ion exchange forces. Because the DNA samples are negatively charged and the nylon membrane is somehow positively charged. So there will be ion exchange forces between the DNA samples and the membrane. And the DNA samples will stick on the membrane. But the water will pass through the membrane because the membrane is a kind of filter. So the water can pass from the membrane and then it will reach the paper towels, which are going to become wet. So I can see that the paper towels are becoming wet, and then I see that the process is working. Now what's going to happen is like this. So this is the gel, this is the membrane, and then when the water is going to pass through the gel to the membrane, the DNA samples are going to be transferred from the gel to the membrane. And then I will have the membrane. What I should do is that I should bake the membrane on 80 degrees of Celsius for 2 hours, or applying ultraviolet radiation to fix the DNA sequences on the.
So both, nitrocellulose and nylon membranes have this property of binding to DNA. Okay, DNA has this ability to go and bind to nitrocellulose or nylon membranes, and then you need to somehow immobilize them because they can float around, right? When you transfer from gel to the membrane, whether it is nitrocellulose or nylon, it gets transferred to the place, but then this is not fixed, it can move around. When you try to do hybridization, it can move around. So in order to immobilize, people do two things. One is to bake them at 80 degrees centigrade for about 1 to 2 hours. Some people do even shorter time, or you can simply expose this nylon membrane or nitrocellulose membrane to ultraviolet light. Okay, and they generally do ultraviolet light for less than 10-15 minutes. Okay, less than 15 minutes actually. When you do either of them, either you can have a UV cross linker, okay, or you can simply leave them at 80 degrees centigrade for 1 or 2 hours. Okay, the DNA gets cross-linked irreversibly. The DNA gets cross-linked irreversibly to the matrix. That means it could be either nitrocellulose or nylon membranes. Okay, and then you can use it for your hybridization.
The membrane, I should perform type of heating. like either using 80 degrees of Celsius or using ultraviolet radiation, like any type of heat. Now, what's going to happen is that my DNA samples will be fixed on the membrane. The thing is that these DNA samples are all double-stranded because when I cut the DNA, I cut it as a double strand. So all the DNA sequences which you are seeing here are double-stranded DNA. So if I'm going to apply the probe, which is complementary to the sequence of my interest, the probe cannot bind to this sequence because this sequence is a double strand. So what I should do is that I should treat the membrane with a denaturation buffer in order to denaturate hydrogen bonds between the two strands of DNA, and then in order to separate the two strands of DNA. So after treating the membrane with a denaturation buffer, all the DNA sequences you're seeing here will be double-stranded DNA. They will be separated. In this stage, I should then apply the probe, which is going to detect the sequence of my interest and bind to it. In order to be able to detect the probe afterward, I should couple the probe with a type of molecule that can give me a signal. This type of molecule can be either a fluorescent, like this fluorescent molecule, or it can be a chemoluminescent. Chemoluminescent is a substrate for a certain enzyme. So if I apply the probe with coupled with a chemoluminescent, I should then treat the membrane with the enzyme. Then the reaction between this enzyme and this substrate will give me a certain signal. Or I can couple the probe with a chromogenic dye, like any type of dye, which is going to give me also a signal. The thing is that this probe is going to detect this sequence and bind to it, and then I can visualize the membrane in a dark chamber, and then I can see the sequence I have. So I can say, yes, I have this gene in this sample, but I don't have it in this sample. So I can see in which sample I have this gene and in which sample I don't have the gene. So this is southern blotting. Sorry, this is southern blotting. Now let's speak about northern blotting. Northern blotting is very similar to southern blotting, but it's used to detect RNA. So let's say I have a cell. This is the genome. These are the RNAs. Then I extract the RNAs, which contain the sequence of my interest. Let's say that this is the sequence of my interest, and I want to detect this sequence on the gel. First of all, as we did in the southern blotting, what we should do is to apply agar gel electrophoresis. Now, maybe you are asking yourself a question: why would I search for a DNA sequence? So let's say if I want to know if this genome contains a several gene or not, why would I search for DNA sequence? Why don't I search for why would I search for the RNA sequence? Why don't I search for the DNA sequence and that's it? The thing is that why we search for the RNA sequence is to see if this gene is expressed or not, or to what extent this gene is expressed. Because in certain type of cells, the gene might be overexpressed or non-expressed. And to see if this gene, so for example, I performed southern blotting, and I see that I have the gene. Now I see that I have the gene, I know that I have the gene, but I want to know if this gene is being expressed or not. Then I search for the RNA, because if I have RNA, then the gene is being expressed. If I have high amount of RNA for this sequence, now I know that this gene is overexpressed. Or if I have a very little amount of the RNA, then I know that this gene is not so much expressed. Because of this, I search for the RNA sequences. Okay. Now, what I should perform is first. First of all, you should know that the RNA sequences exist always in a secondary structure. So the RNAs are always in a secondary structure. And if I apply the probe, if I design a probe which is complementary to this sequence, and I apply the probe directly on the membrane, the probe cannot bind to this sequence because this sequence is a secondary structure. So what I should do first is to denaturate the secondary structure of the RNA to produce a linear structure of the RNA in order to be able to bind the probe with the RNA. To do so, I apply agarose gel electrophoresis, but with a small addition, which is the addition of formaldehyde to the gel. So what I do is I add formaldehyde to the gel, and formaldehyde will be responsible to denaturate secondary structure of the RNA to a primary structure, which can be then detected by the probe. Let's say that these are the samples separated. Then the secondary structure of the RNA will be denatured to a primary structure, which can be then detected by the probe that I designed for this sequence. Now, similarly to what we saw in Southern blotting, let's say this is the sequence of my interest. We should first transfer the RNA sequences from the gel to a membrane, and the process is the same what we saw in Southern blotting. So first we use the chamber, we use an alkaline buffer, a sponge, the gel, a membrane. Here we should use RNA binding membrane, which is the same. It's the nitrocellulose membrane, paper towels, and weight. Similarly, the capillary forces are going to drive the water from the area of high water pressure to the area of low water pressure, and then the RNA sequences are going to be to stick on the membrane because of ion exchange, because of an ion exchange forces, because the RNAs are negatively charged, and the membrane, the nylon membrane, the nitrocellulose membrane, is a type of positively charged. So then I will have the RNA samples stuck on the membrane like this. Then I can apply the probe on it. The probe here is an RNA sequence, so we have uracil instead of thymine. And yes, this is the probe. The probe should be also coupled with either with a fluorescent or with a chemoluminescent using an enzyme, or with a chromogenic dye, the same what we used in Southern blotting. And then this probe is going to detect the sequence of my interest and is going to bind to it. Then I can visualize this sequence in a dark chamber, and I can see how much RNA I have in this sequence. Then I should know if this gene is being expressed or overexpressed or not expressed. This is everything I wanted to tell you about northern blotting and southern blotting. I hope you enjoyed.
So, one important thing, application of northern blot is for quantifying your specific messenger RNAs. Okay, this is very, very relevant for when you compare the expression pattern of genes between, say, a normal cell and a cancer cell. A cancer cell has so many different, genes that are not expressed at all, and there are certain genes that are overexpressed, right? So you can essentially, you can do a quantitative test. Okay, by doing a northern blot, you can figure out whether a gene of interest, is getting overexpressed or not, or is it getting underexpressed or not. For example, most of the oncogenes are expressed at a very high level, okay, in a cancer cell. On the other hand, tumor suppressors are expressed at a very low level, okay, in a cancer cell. In a normal cell, they have a certain level of expression, but in a cancer cell, oncogenes are expressed at a very high level and tumor suppressors are expressed at a very low level compared to a normal cell, which you can easily quantify using this technique. Okay, let us look at another one, little more detailed explanation on southern blot. Give me a minute. Before that, I saw two videos, one from High Media, which is this thing, and maybe we will see the first one. and then maybe look at the other one also.
Everything expressed here is just my own opinion. Look, I thought I'd share with you all something that I learned many, many years ago,
As soon as
and something
I
that
will.
was always a wonder to me, and one of the things that really kept me going and studying molecular biology over the years, just becoming fascinated with nucleic acid hybridization. I'm going to show you, give you all a brief history of the miracle of nucleic acid hybridization, how it was used back in the day. Let me introduce you all to Sir Edwin Southern. This is a guy who knows how to solve a problem in a very simple fashion. I'll let you know what he did in 1975. Let's get into what he did in 1975. He invented something called a Southern blot, all right? And it has to do with putting DNA onto a solid matrix so you can work with it. You can do some experiments with it.
त्योहार की फैशन शॉपिंग only with Myntra Big Fashion Festival starts 3rd October. Don't miss out.
Don't try this at home. Try LiveSpace. Design your home interiors with better quality materials and fittings. LiveSpace your space.
So first thing you got to do, Southern blotting and the power of base pairing hybridizations, you got to acquire some sort of DNA sample. Southern blotting involves DNA. There's another example called Northern blotting. Somebody was having fun with names there, and they called it Northern blotting, and it has to do with RNA. But right now, let's talk about DNA. You acquire some sort of DNA sample, whatever it is that you're studying, and you purify it some way. We used to use proteinase K, that's PK, and SDS, which is a detergent, and do what's called phenol chloroform extractions. It's very messy, but it's a lot of fun. And you do an ethanol precipitation afterwards. Or you can do what's just called the Kaizen kits. Kaizen always makes the best kits. You can purify DNA that way. And you have to run your DNA sample in some sort of gel. This is an agarose gel. This is a small horizontal gel apparatus. Notice it's got a power supply, and it's got a negative pole over here with the black lead, and it's got a positive pole with the red lead. The DNA is always loaded at the negative pole because it always wants to run towards the positive pole. Reason being is because the net charge on DNA is negative because of the phosphate groups. Right? We used to have, I mean, in my labs, we used to have just massive gel boxes to run, 100 samples at once or whatever. And we had small ones too, like this one, but the big ones are a lot of fun to play with. And here's what some genomic DNA from whatever cell it's from, whatever organism it's from. Here's what it looks like after it's run in a gel. So here's your negative pole. Here's your positive pole. The DNA is running down here. Whoops. Screwed up already. DNA is running down. So the first line here is just a marker, a DNA ladder it's called. The first one is undigested genomic DNA. And it's by digested, I mean it was not cut with a restriction enzyme. It was taken from the cell, purified, and thrown into the gel. And it's not a lot of DNA. It's not fireballing, so it's probably somewhere between 50 and 75 nanograms, maybe 100, although I doubt it's 100. Because this gel looks to be, this is not one of mine gels. I just picked this off the internet. But the next lane over here is genomic DNA that's been digested with some sort of restriction enzyme or multiple restriction enzymes. And you can see they just repeated the thing over here. So how you visualize the DNA is actually, well, this is run in a 1% agarose gel, as I said, in some sort of buffer. We use tris boric acid or tris acetic acid, EDTA buffers. And we sustain it with lithium bromide, and it intercalates between the base pairs. Shine a UV light on it, and it will fluoresce, as you see here. I'm sure there's stuff they use now these days that are not as toxic as lithium bromide. But it's really cool. I spent half my life under UV lights taking pictures of gels. It's really neat. Not half my life. Slight exaggeration, but you get the point. So then you prepare your DNA for hybridization. And the whole idea behind hybridization is you take some sort of DNA probe and you stick it in there so it will hybridize with your DNA of interest. In order for the DNA to be hybridized to, you have to separate it. You have to denature it. And we used to do that with sodium hydroxide. So we would simply soak our gel in sodium hydroxide and salt for about 15 minutes or so. And the 500 millimolar sodium hydroxide would denature the DNA. And afterwards, you've got to neutralize the sodium hydroxide. And you do that with an equimolar amount of tris, always keeping the salt present to keep the DNA happy for about 15 minutes or so on the rocker, and that neutralizes it. Then you proceed to blotting. And here's what Southern did, which is just brilliant. First thing you got to do is find the lasagna dish, right? And so here's what a gel looks like. That's after the blotting, but so you make some sort of support inside the lasagna dish and you fill it with buffer. You got some buffer here. TBE's fine, TAE, whatever you got hanging around, whatever's in the gel, you know. And then whatever you ran the gel is fine. And then sponge, you put an actual, just a sponge. And then you have what's called Wattman filter paper. That's the black bar here. It's just a piece of paper. They call it filter paper. And then you've got an agarose gel. That's your gel right here. Here it is. There's your agarose gel. And on top of that, you have your nylon or nitrocellulose membrane sticking right on the gel. And then another piece of Wattman filter paper. And then you build what's called the parafilm frame all around it. And then here's the high-tech stuff that Edwin Southern thought about. You put paper towels and a weight on top of it. Isn't that brilliant? So what happens is that the buffer, via the dry paper towels and the weight, capillary action simply draws the buffer up through the sponge, through the agarose gel. The DNA, there's enough force there to take the DNA out of the gel, and the DNA gets stuck onto your membrane, the nylon or the nitrocellulose membrane. All right? And then you do that at night when you go home. In the morning, 16 hours later, the paper towels are all wet. All right. A lot of the buffer has been drawn up. You disassemble the apparatus, and your membrane looks just like this. It now has the DNA on it, and that DNA is denatured. Remember, because you treated it with the sodium hydroxide. Then you have to permanently affix the DNA to the membrane, and how we used to do that was we used to hit it with 50 millijoules of UV in what's called a crosslinker machine, and the 50.
So in the previous one, they said that we can bake it at 80 degrees centigrade. Either way, as I said, there are two ways by which you could cross-link the DNA to the membranes, either by heating them in an oven at 80 degrees centigrade, or by cross-linking them using UV.
50 millijoules was enough energy to form what's called cross-links. So the DNA gets cross-linked to the membrane so it won't move around. It stays put, basically. Here's what you do next is you make what's called a probe. We used P32, which is radioactive, of course. And we would make a P32 DNA probe or an RNA probe, which is called a riboprobe. It doesn't matter. Riboprobes are hotter. They are really smoking hot. What's called random prime DNA probes are not as hot, but they're still good. But riboprobes was always the way to go. They're just smoking hot. That is an RNA. This all comes back, right, to CRISPR-2. If you make a riboprobe, that's an RNA. And if you put it on your Southern blot, the RNA is going to hybridize with the DNA on the blot, right? That's like the CRISPR guide RNA hybridizing or finding the target inside the genome, right? So you make a radioactive probe, DNA or RNA probe, and then you clean it up with some of these columns to get rid of the unincorporated P32 because you don't want those little things hanging around. And, some of you may ask, well, where do you buy the P32? Where do you buy the radioactive stuff? Well, here's from PerkinElmer, right? This stuff right here, I got this picture and this information from PerkinElmer's website. So, for example, you would buy deoxy, GTP, guanosine triphosphate, and it says alpha P32. So the alpha P32 would be the first phosphate from the triphosphate. That's the one that's closest to the guanine, right? And they tell you how hot it is. This is 3,000 curies per millimole. It's, you know, the concentration is 10 millicuries per mil, and they give you 250 microcuries of it. All right, so you buy this stuff straight from PerkinElmer, or a lot of places sell it. All right, so you now have a smoking hot riboprobe that's labeled with P32 or a DNA probe. What we used to do is we had these bag sealers then. All you do is you combine your blot, your DNA blot, your P32 probe, your radioactive DNA or RNA probe, and you put it in a bag. We used to use bags. Some people use glass tubes. Put it in a bag with some buffer, and believe it or not, we used what's called salmon sperm DNA to block the hybridization overnight. I know what you're saying. You're like, is that what I think it is? Yes. It's sperm from salmon. And we used to buy truckloads of it. Not truckloads, but, you know, milligrams of the stuff. And that was a common blocker used in hybridizations. You would stick it in with your hybridization, and it would block non-specific sites. So inside the bag, you've got your buffer, your salmon sperm DNA, your blot, of course, that's got your target on there somewhere, and your P32 probe. You seal it up, you hybridize it overnight in a water bath, and then you put it in a box. You put it in a film cassette, right? So you hybridize it overnight. The next morning, you place it inside the film cassette. You cover the blot with X-ray film, and you expose it for the desired time. And we used to expose ours at minus 80 degrees centigrade. Then you develop the film. Here's what one of these things look like. This is an actual northern blot, but it's the same. It's not a southern blot, but it's a northern blot, but the procedures are the same. Instead of running DNA in a gel, I ran RNA, right? And these were with P32 probes. So in the lane here is total RNA from the cells. The thing that always amazed me when I first did one of these.
So you can imagine, I said northern blots can be used to quantify the extent of gene expression. How much RNA is there. You can actually see very clearly between this and something like this. There is a huge difference in the amount of RNA that is getting expressed. Okay, so this means there is a very high level of expression. This means there is a bare minimal expression, right? And this is used as a control. This is not supposed to change at all. So when you do northern blots, southern blots, always use certain controls. These are called as housekeeping genes whose expression does not vary. So that you can say that, this is not an artifact, but this is an experimental one. Suppose you also observe a similar thing like this for GAPDH, then that means your experiment is not right. You will have to repeat the experiment. But the fact that, these housekeeping genes are not showing any difference in expression, but these two are showing differences in the expression, tells you that the RNAs are getting expressed at different levels.
These things years ago when I first did a southern blot is that you can take a probe, stick it onto a membrane, let it go overnight, and that probe, whether it be DNA or RNA, will find that target on that blot among the millions of other targets. Isn't that amazing? And so what you're seeing here, the black band, represents the amount of RNA that's in that particular lane, right? So for example, here's my reporter I had, right? So if I had a reporter, this is an actual experiment where I treated cells with the vehicle, V. I treated them with a heavy metal cadmium or with something called hemin. And you can see this particular gene went up. As the band gets darker, the RNA, that means that there's more RNA targets on the blot, which means that the gene was turned on more, or that the reporter in this case was turned on more. And here you can see it was turned on a lot, and a lot here. And here you can see with this particular deletion, or this particular mutant that I had, the M3, the induction of the reporter was almost completely wiped out. And here's the endogenous gene right here. The gene's called hemoxynase 1, but you can see the endogenous gene. And this bottom one here is a loading control to prove that there's an equal amount of RNA in each gel. So this is actually one RNA blot, one northern blot that I probed 3 different times. I probed it with this reporter, I probed it with this gene, the HO1 gene, and I probed it with this um, gene. What's the word I'm looking for? I probed it with this probe, the GAPDH probe. Here's the HO1 probe, and here's the HGH reporter probe. This is all done before I had real-time PCR. Nowadays, you do this in a real-time PCR. All right, you can do it, in an hour and a half in real-time PCR. Back then, you had to do northern blots, and I don't know. To me personally, I kind of like doing northern blots. Here's another northern blot I thought I'd show you. This all is a developmental northern blot of a gene called E23 in Drosophila. This is from my first publication, and this is really cool. This is the E23 gene during the developmental time course of the organism, from right when the egg is laid, over here in the first lane, to over here when they emerge from the pupa case as adults, male or female. And you can see how this gene is tightly regulated through time. It's regulated by a hormone called ecdysone. Every time you see these black bars, that's when the ecdysone is turned on inside the organism. So this is the power of hybridization. And of course, the nucleic acid interaction between the guide RNA of the Cas molecule and the target is what makes CRISPR possible, right? It's what makes base editing possible. It's what makes prime medicine possible. It's what makes the epigenetic CRISPR stuff possible. I never thought I'd be looking at this stuff again. I did this stuff so many years ago, and I just never thought I'd be looking at it again. And then, boom, CRISPR comes along, and I start getting into it. Like, I don't know, 5 or 6 years ago, I started reading about it and trying to understand it. Started really getting into it like 3 years ago and started buying stock. And I never thought that all this hybridization stuff that I had experience in years ago would come back and like, oh my God, this is CRISPR. This is what CRISPR. And then overnight, just overnight, among the millions of target molecules on that blot, your probe will find it and it will stick to it and it will light up on an autorad, just like this, cleanly and specifically. It's always amazed me. No wonder CRISPR works. I wanted to share this with y'all. Anyway.
Okay, so tomorrow we will see how this now has been developed as a kit. You can basically do this even at the school level, right? So High Media, which is one of the company that makes all these medias for bacterial growth, they have a teaching kit. Maybe we'll see. If possible, we'll see. Otherwise, I will share the YouTube link with you. You could have a look at it. Okay. So basically, the hybridization happens because of complementarity, right? So when you use DNA to DNA, it is Southern blot. When you use RNA and DNA, right? In one of the things, RNA is going to be the this thing, right? If that is the case, then it is called as a northern blot. Okay. DNA hybridizations are typically much more easier to do. The RNA has this ability to degrade very quickly. So many people who do northern blots only if necessary. It's very informative, no doubt about it. It's little tedious, and nowadays with real-time PCR doing such a wonderful job, people generally don't go for northern blots, right? So these are historical techniques that I thought I should, all of you should have knowledge of, based on simple hybridization kinetics, complementary hybridization. You go for southern or northern blots. Okay. We'll take up the rest of the things tomorrow. Okay. Any questions?
No, sir.
no questions, we'll meet tomorrow. All right. Okay. Take care. Bye.
Thank you, sir.
Okay.